Dear children, I am here with another video of solving biology sample paper. So this time I have taken the sample paper 1. So let's get into the solutions of this paper. Yes children, I have taken the sample paper 1. Let's see the solutions. Section A. If most individuals in a population are young, why is the population likely to grow rapidly in the future? You guess which chapter? Yes, organisms and population. So what are they talking about? Actually, they are talking about this age pyramid, okay? So did you get that? What are they talking about? If most individuals in a population are young, why is the population likely to grow? So they are talking about the growth pyramid or age pyramid. And let's see the options. Many individuals will begin to reproduce soon. Death rates will be low. Immigration and immigration can be ignored. All of this. So they're talking about the expanding population. Let's check the expanding age pyramid. In the expanding age pyramid, you can see the lowermost or the base one is includes the pre-reproductive age group, right? So in that, if that case, like pre-reproductive age groups comes as the base of this age pyramid, then there is a chance of reproductive ages in the coming generation, right? In the coming period of time, so that the population will be expanding. Just consider the other two cases. In this stable age pyramid, the pre-reproductive age group is less. In that case, the growth of the population will not be expanding. It may be stable for a period of time. And the last one, if the pre-reproductive age group is less, then the age pyramid shows a declining growth. You got it? So now you can guess what the answer shall be. Yes, the first one. Many individuals will begin to reproduce soon because it is pre-reproductive. Fine. So this shall be the right option. Now next one. Primary endosperm nucleus, PEN. Keep that abbreviation in your mind. PEN stands for primary endosperm nucleus. And the cells formed from that is PEC, primary endosperm cells. You remember which chapter it is from? Yes, it is from sexual reproduction in flowering plants. Let us get into the chapter. Yes, sexual reproduction in flowering plants. They are talking about the primary endosperm nucleus. So where does it come? Primary endosperm nucleus. It comes under the fertilization, double fertilization, triple fusion, that part. Okay. So after the structure of embryo sac, synergids, all the structures, pollination, followed by the fertilization process. It comes in the fertilization process. Yeah, you can see here. And under the heading double fertilization, the question is like this. Primary endosperm nucleus, PEN, is formed by the fusion of, you can see that here. The other male gamete, the second male gamete moves towards two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus. So it's triploid, triple fusion. Okay. As this involves the fusion of 3 haploid nuclei. How did we get 3 haploid nuclei there? The second male gamete and the 2 central polar nuclei, which are haploid. So 3 haploid nuclei fuses to form the triploid primary endosperm nucleus. So let's check the question. What are the options given in the question? 2 polar nuclei plus 1 synergid cell nucleus. No. 1 polar nucleus, 1 antipodal, 1 synergid. No. 2 polar nuclei and 1 male gamete nucleus. Yes, that's right. 2 antipodal cell nuclei and 1 male gamete nucleus. So that's also not the one. 2 polar nuclei and 1 male gamete nucleus. Okay. Next question. Which enzyme helps in removing oil stains from clothes? Can you guess which chapter it is? Microbes in human welfare? Yes. Industrial uses of microbes in human welfare. Let us check the chapter. Microbes in human welfare, you can just see under chemicals, enzymes and other bioactive molecules. You can see here, yes, here the sentence comes. Lipases are used in detergent formulations and helpful in removing oil stains from the laundry. Okay, did you see that? Please mark it in your textbook. So the answer is lipase. Option C, lipase is the answer. Next question. Which of the following cause transmission of HIV, multiple sexual partners, transfusion of contaminated blood, sharing infected needles, all of this. Yes, it's all of this. It can spread through multiple sexual partners, or by transfusing contaminated blood, or by sharing infected needles. So the answer is all of this. Next question. Hardy-Weinberg equilibrium is known to be essentially affected by the factors like gene flow, genetic drift, mutation, genetic recombination, and those who have learned the Hardy-Weinberg equilibrium and the statements followed that will be knowing the answer well. If you do not know, let us look into the textbook and confirm what the answer is. Let's go into the textbook. So which chapter it is? Hardy-Weinberg equilibrium? Yes, it is evolution. Let's take the chapter evolution. Let's get into the page where the Hardy-Weinberg's law comes. Hardy-Weinberg's principle. You can see a statement given here. Hardy-Weinberg equilibrium. These are 5 factors are known to affect Hardy-Weinberg equilibrium. These are gene migration or gene flow, genetic drift, mutation, genetic recombination, and natural selection. So these are the 5 factors that affect the Hardy-Weinberg equilibrium. So the answer here is natural selection. Next question. Plasmid used to construct the first recombinant DNA was isolated from which bacterium species? See, this is a bit confusing question. Many children used to write it as E. coli. And do you think that it is E. coli? Escherichia coli? Shall we check the answer? Yes. Let us get in the chapter. Biotechnology. Biotechnology principles and processes. You can see here, towards the beginning of the chapter, under the heading Principles of Biotechnology, you can see here in this paragraph, they're talking about plasmid for the first time. So the construction of first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid, autonomously replicating circular extrachromosomal DNA. So that is the definition of plasmid. Autonomously replicating circular extrachromosomal DNA, and it is first isolated from the bacteria Salmonella typhimurium. Remember, Salmonella typhimurium. Okay, so the answer is Salmonella typhimurium. Is that from that bacterium the first plasmid was extracted. Now, next question. Microbes are used in, so this is obvious that it is from microbes in human welfare, sea-based treatment, isn't it? So, microbes are used in primary treatment, secondary treatment, anaerobic sludge digesters, production of biogas. So, 4 steps they have given here. Which are the steps do we use or do we include bacteria in? Any idea? Guess the answer. We'll check it from the textbook. Microbes in human welfare. And you know it is in sea-based treatment. Yeah. So, primary treatment. Here, do they use the bacteria or the microbes? These treatment steps basically involve physical removal of particles. So there is no role of bacteria or microbes here. Coming to the secondary treatment, the primary effluent is passed into large aeration tanks where it is constantly agitated and aerobic microbes are allowed to grow. So secondary treatment, we use aerobic microbes. Then after that we get the sludge. So we use anaerobic bacteria as the sludge digester. So there also we use, and we know for the release of biogas also microbes play a major role. So let's see the question. Primary treatment, no. Secondary, yes. Anaerobic, yes. Production of biogas, yes. So what will be the answer? Option C, 2, 3 and 4. Clear? Next question. If a double-stranded DNA has 20% of cytosine, what will be the percentage of adenine in it? In the previous question paper which we have discussed, we come to know we have checked the Erwin-Chargaff's rule. Do you remember? So let us get into that rule again from the textbook. Molecular basis of inheritance. In that, under Erwin-Chargaff's rule, you can see here under the structure of polynucleotide chain, they are talking about Erwin-Chargaff's rule, where he states that the ratio between adenine, thymine, guanine and cytosine are constant and equals 1. So this can be interpreted in 2 ways. So we'll get into the question. This can be interpreted like this. That is, adenine will always be equal to thymine and number of guanine will be equal to cytosine. So it can be written like this: adenine plus guanine will be equal to thymine plus cytosine. That is, number of adenine and thymine will always be the same, and number of guanine and cytosine will always be the same. So here they have given that 20% of cytosine. So cytosine will be 20%, right? And therefore guanine will also be 20%. So the total nitrogenous bases, if you take adenine, thymine, guanine and cytosine, is 100%. Then guanine and cytosine 20%, so it is 40%, and rest will be 60%. And we know that adenine equals thymine, so 60 by 2, it is 30%. So the answer is 30%. Adenine always equals to thymine, guanine equals to cytosine. So adenine plus guanine will equal to thymine plus cytosine. And if you know the number of guanine or cytosine, we know both are the same. So together it forms a 40% of the nitrogenous bases, and rest will be 60%. And out of that, half of it goes to adenine and half of it goes to thymine. The percentage of adenine is 30%. Hope it's clear to you. Then next question. The density of a population in a given habitat during a given period fluctuates due to the changes in certain basic processes. On this basis, fill up boxes A and B given in the flow chart. So those who have learned this flow chart diagram, it should be really easy. It's just a direct question. So let us recollect the diagram from the textbook, Organisms and Population. Let's see that flow chart. Yeah, it's here. So they have given the horizontal one. So this, when this box was considered as A and this box was considered as B. On the basis, fill up boxes A and B in the given flow chart. So A is increasing. When A enters, it increases, the population density increases, so it adds to the population density. And B, when it goes out, it reduces the population density. So A will be natality rate and B would be the mortality rate. So let's check the question. A natality and B mortality. Okay, so option A. Is it clear? Natality, mortality. Next question. The given Punnett square represents the pattern of inheritance in a dihybrid cross where yellow and round seed condition is dominant over white and wrinkled seed condition. So here better you have to solve this cross, shall we? So it will be Y, Y, R, R. Okay, now the plant of type H, so this is type H, okay, will produce seeds with a genotype. So what is the genotype? Capital Y, small y, capital R, is identical to the seeds produced by the plants of, so this is identical to which one? Capital Y, capital R, small r. This is the identical one, right? Yeah, they're talking about the genotype, children. Genotype stands for the type of genes, the alphabets. Okay, so those are not able to remember what the genotype and phenotype is, genotype always stands with the alphabets what we use for, and phenotype is the morphology, or what character do they express, what trait do they express. That's what phenotype refer. Here they have asked for the genotype. So the genotype, this both are identical. So the answer is type N. I hope you understood how to solve this. And if children if you are able to solve without writing this table, that's quite fine. And if you are finding it confusing, please solve the table and then write it. It will not take much time for you to do this. Now, match column 1 with column 2 and select the correct option from the given codes. DNA recombinant technology. What is DNA recombinant technology? Yes, it is genetic engineering. We know it is genetic engineering. Precipitation of DNA. What do we use here for precipitating? That's pooling process. We use chilled ethanol. PCR, polymerized chain reaction. What we do is actually amplifying the number of genes. So gene amplification. And ethidium bromide, we are using it in gel electrophoresis method. which help us to view the DNA fragment. So DNA staining, actually only by the use of ethidium bromide, the UV rays will show it. Okay, we use the combination of ethidium bromide and UV light to view the DNA fragments clearly. So DNA staining. So which option stands for this match? A4, B1, C3 and D2. Yes, option A. The given table shows differences between spermatogenesis and spermiogenesis. Select the incorrect option. So spermatogenesis is the entire process, right? And the differentiation of spermatid to a spermatozoa, we call it as spermiogenesis, the last process. You remember which chapter it is? Yes, human reproduction. Now, second point. So the first point seems right. Second point. It changes a haploid structure to another haploid structure. No, actually spermatogenesis includes the meiotic division. That is, sperm mother cells will undergo meiotic division and then forms the haploid cells. Whereas spermiogenesis, there is no conversion of haploid to, diploid or haploid conversion is not there. Already meiosis is converted and that haploid to haploid structure, haploid thing is differentiated into another haploid form. So second statement seems to be wrong. Let's see the next one. Growth and division occur. Yeah, it's right. Divisions and growth are absent. Yes, there is only differentiation. A spermatogonium forms 4 spermatozoa. Yes, from the entire process, a spermatogonium forms 4 spermatozoa. And here, a spermatid forms single spermatozoa. And it is just differentiation, right? So, a spermatid differentiates into spermatozoa. So, this statement also seems right. Only statement B is wrong. So, they have asked for the incorrect option. So, option B. Coming to question number 13. Elimination of competitively inferior species in a closely related or otherwise similar group is known as competitive exclusion principle. Yeah, competitive exclusion principle comes under the interaction or the relationship. Shall we get into that chapter? Yes, organisms and population. Organisms and population. We'll go for the interaction, population interaction. And that when it comes to competition, there is a principle called competitive exclusion principle, Gauss competition exclusive principle. So this principle states that two closely related species competing for the same resource cannot coexist indefinitely and competitively, and the competitively inferior one will be eliminated eventually. That is, two species are competing, two closely related species are competing for the same resource, and they cannot coexist because they cannot coexist indefinitely because the competitively inferior will not be getting that resource, and there is a chance for them to get eliminated. So that's what Gauss competitive exclusion principle states. Let's see the question again. Elimination of a competitively inferior species in a closely related, otherwise similar group is known as competitive exclusion principle. Yes, this statement is correct. Assertion is correct. Assertion is true. If two species compete for the same resource, they could avoid competition by choosing different times for feeding or different foraging pattern. Actually, the second statement, reason, what they say is a solution for this Gauss competition principle. Let's get back to the chapter again. See the next sentence. This may be true. That is, this competition exclusion principle may be true. That is, inferior species may get eliminated if resources are limiting. If there are only limited resources, this may happen. But if the resources are unlimited, there's no chance for this competition, right? The inferior one also will be getting the resources. So, not otherwise. Again, more recent studies do not support such gross generalization about competition. They say that recent studies, they don't support this principle, exclusion principle, saying that while they do not rule out the occurrence of interspecific competition nature, they point out the species facing competition might evolve mechanisms that promote coexistence. Though they are inferior, they'll find some other way to survive. That's what the recent studies teach us. One such mechanism is resource partitioning. What they do is, if two species compete for the same resource, they could avoid competition by choosing different times or different foraging pattern, different feeding patterns, or different eating pattern or hunting pattern. So let's get back to the question again. If two species compete for the same resource, they could avoid competition by choosing different times for feeding or different foraging pattern. Yes, reason is also stating a true fact, isn't it? So assertion is true, reason is true. But you think, is reason explaining assertion? No, reason is talking about another chance for stating that assertion may not be always true, isn't it? So you can go for the option B. Both A and R are true. A is true, R is also true, but R is not explaining the statement A. They are giving another option for A, statement A. So A and R are true and R is not the correct explanation of A. Coming to question number 14. Mouse is the most preferred mammal for studies on gene transfer. Mouse possesses features like short estrous cycle, gestation period, relatively short generation time, production of several offspring per pregnancy, etc. Yes, that is the reason we have chosen mouse as the most preferred mammal for study on gene transfers. So option A, both A and R are true and R is the correct explanation of A. Now coming to the next question, 15. Given below is a diagram of a normal 28-day menstrual cycle in a human female. It depicts phase 1, 2 and 3. Okay, phase 1, 2 and 3. So phase 1 shall be the menstrual phase, 1 to 5 days or 6 days. And then the next one is luteal phase, and next comes the follicular phase, right? Okay. And study this diagram and comment upon the appropriateness of this assertion and reason. The hormone secreted in large amounts in phase 3 is responsible for maintaining pregnancy. Yes, during phase 3, the follicular phase, Graafian follicle ruptures, it forms corpus luteum and it secretes progesterone more during this phase, isn't it? So it is responsible for maintaining pregnancy. Now, so statement A is true. Now, corpus luteum secretes progesterone in phase 1. No, in phase 1 actually estrogen is more. However, it degenerates completely in phase 3. That's also wrong. It secretes progesterone in phase 1 is wrong. So, A is true but R is false. Okay. Now, next question. Emigration is outward movement of some individual from local population. Yeah, next question. Emigration is the outward movement of some individuals from the local population. Yes, emigration is moving outward, right? Now, emigration is caused by the occurrence of deficiencies and calamities. Yes, people move out due to it can also be caused by the deficiencies and calamities. So, both A and R are true and R is the correct explanation of A. Now, coming to section B.